Okay, so what we were discussing about this is now the part two of this lecture continuation that HIV infection compromises host immune system. So basically, what are the target sites where this HIV virus can attach, when where they can grow? What are those specific cells? So these HIV viruses they target CD4 cells, mainly helper T cells, and without these cells, the problem is that the host body or the human body cannot mount any effective immune response. So that is the problem when HIV infects these cells and gradually the number of these cells goes down. The overall immune system of the human body gets deprived, and as a result, the multiple infections that usually in normal routine does not cause that much problem, they started to cause problem, and variety of opportunistic infections can also start, and these are the one that cause the disease, the person to die, not the actual HIV problem. In the patients with the HIV infection, clinical symptoms usually appears after 8 to 10 10 years. It means that this virus can remain dormant in the host cells for that much long period of time, and during this period it gradually integrates into the host cell DNA and multiply with that host cell and gradually increases in number, in fact more and more cells, and that's how that after that long period of latency, all of a sudden the patient starts to have more symptoms appear. For the testing of this disease, we are not going to look for the virus itself. What we look for, if, especially for the diagnostic purpose, we look for either the antigens. These antigens are those specific cell surface markers that I have shown you on the envelope viruses. So these are the GP120 proteins that are found on the surface of the envelope of the HIV viruses. These GP120 proteins they serve as antigen, and they specifically attaches with the CD4 surface receptor markers that are present on our T helper cells. So once we have this attachment happen and gradually the body of the humans started to recognize these the presence of these HIV cells in the overall circulation, some of our immune cells they also starts to produce antibodies against these viruses. So there are two things. One is the antigenic marker that is present on the surface of the envelope of the HIV virus, that is GP120, that we are looking for in the blood and other secretions. Beside that, the antibodies that are produced by the human body in defense against this virus. So these two things we are looking for in the patients for the presence of this infection. How this infection spreads? So the carriers usually they do not have clinical symptoms for that long period of dormancy, but because the virus is there in their body, so they are infectious too, and they can transfer this from one person to other person. Infection continues throughout this latent period, and also during the integration of the DNA or the viral DNA into the host DNA, mutations are also happening. So these mutations allow the virus to overcome immune system, and that's how the AIDS starts. Uh, there are a variety of animal viruses, and for every single virus, there are different models that we have in consideration. Like for example, this influenza virus or the coronavirus or the flu viruses, they need the host respiratory cells to proceed and to increase in number or to survive. While on the other hand, for the HIV cells, we see that the virus needs the T helper cells or the macrophages that have particularly CD4 receptor cells, and that's why we say that this HIV virus only attacks the CD4 positive cells. What happened actually that the viral GP120 proteins that are present on the viral envelope, they attaches with the CD4 proteins on the macrophages and the CD4 cells, and after that they integrate the or they inject the viral nucleic acid into the host cell. So here you can see this whole mechanism that how it is going on. So you can see here this is the HIV virus having these GP120 proteins, glycoproteins, and on the other hand this is the human macrophage or the T helper cell that has these CD4 receptor cells. So the virus attaches with the CD4 receptor T cells on the CD4 receptors on the T cells by means of these GP120 proteins, and once this attachment happens, the nucleic acid from the virus enters the host cell. The virus also is the virus we are particularly talking about the HIV virus. It is also RNA double-stranded RNA virus, so the RNA cannot serve as it is as a DNA template. So first thing that this virus need to do is to make the DNA by using this RNA as a template. So with the help of the reverse transcriptase enzyme that this virus brings with it RNA into the cell, it will make DNA using this RNA as a template, and then this DNA will go into the nucleus of the host cell where it will integrate into the host DNA. Once it is this integration is complete, the normal routine procedure will start. That is, the DNA will make the messenger RNA by means of transcription. This messenger RNA will go out into the cytoplasm, and with the help of the ribosome and the messenger RNA and the tRNA, transfer RNA, and the ribosomal RNA, it starts to do the protein synthesis. Now, what actually is going on in this protein synthesis is that the proteins beside the host cell proteins, the viral proteins and viral RNA are also made, and these raw materials are gathering into the host cell cytoplasm. And once enough of these raw material is there, the assembly of the viral particles will start. So you can see here that viral capsid and the nucleic acid assembles together, and this assembly of the nucleocapsid will go out of the viral host cell by means of budding, and when it happens, this part of the host cell wall becomes the envelope of the virus. So the virus will become the complete HIV particle having an envelope that has the GP120 specific virus specific proteins, a nucleocapsid, and the RNA that is the nucleic acid material of the virus. During an infection, HIV is constantly replicating and mutating, and eventually virus mutates to infect a broader range of cells, leading to a loss of T helper cells. And this destruction of T cells, it blocks the body's immune response, and it may result in cancers and opportunistic infections. For the treatment of the disease, as we have seen that what are the different stages of this viral replication and into the host cell. So like we have the one area where we can stop this is by blocking this entry of the virus into the host cell. Second, if the entry happens, then we can try to stop the release of the nucleic acid into the host cell, so by blocking the entry by using fusion inhibitors. If the entry happens or the viral nucleic acid enters the host cytoplasm, then we can use the reverse transcriptase inhibitors that can stop the replication of this RNA into the DNA. If this happens, okay, in next stage is the integration of the viral DNA into the host DNA. So we can stop that by using the integrase inhibitors. Then if we are still not able to stop this virus, then after the integration there is one more stage that we can still try. That is how we can stop the synthesis of the proteins and the other raw materials. So by using the protease inhibitors, we can stop the processing of the
protein synthesis and the viral protein synthesis. So these are the different mechanism that we can use to overall inhibit the viral entry. And FDA it has lists about 34 antiretroviral drugs that are used in AIDS therapy, and most of them are targeting any of these or sometimes some of these collective target sites. Out of all those treatments that are available, AZT is the first drug that was licensed for the clinical use. Basically, it is a reverse transcriptase inhibitor. So, what it do? It basically stops the transfer of RNA, that is the HIV RNA, into the DNA. Beside that, we have variety of protease inhibitors also that stops the synthesis of the proteins for the virus. There are some fusion inhibitors also available that blocks the viral entry into the host cell, and that's how we are trying to overcome this problem of AIDS. Beside these, also we use the combination therapy. So the combination of drugs has entirely eliminated the HIV virus from many patients and their blood streams, and it is called as HART, that is highly active antiretroviral therapy, and it has cut the US AIDS death rate by three-fourths since its introduction in the mid 1990s. But the problem is that we have to keep the treatment continue. If we stop the treatment, the virus levels might rise again. So, with reference to the development of the vaccine against HIV, what are the problems? So, the problem is mainly the high mutation rate. So, when the virus enters the host cell and it is converting into DNA from RNA by using the reverse transcriptase enzyme, that is the time when the most of the mutations happens. So real problem is not the vaccine yet has produced a strong cellular immune response, and that is why we are still not able to provide any vaccine for this. Some of the attenuated simian immunodeficiency virus vaccines have been also tried in animals, but it is still under process. Another important viral infection is influenza. That is one of the most lethal virus in human history before corona. Influenza pandemic of 1918 and 1919 thought to have infected one-third of the world's population. Again, same as your HIV virus, flu viruses are also annual viruses. There are three types of these flu viruses depending upon the based on the capsid proteins: type A, type B, and type C. Type A is considered as more severe kind of flu virus as compared to the type B and type C. Um, they have lots of subtypes also that differs in protein spikes. The most common protein spikes, like we have GP120 in case of AIDS virus. So in case of influenza virus, we have two type of these proteins present on the surface of the envelope of influenza virus. These are called hemagglutinin and neuraminidase, and both of these antigens, they have a difference in their mode of action. So hemagglutinin, as the name indicate, heme for iron, and agglutinin that helps in the attachment. So it basically aids in or helps in the viral entry, while neuraminidase is an antigen that helps in the viral exit from the host cell. So these HNN molecules, they basically accumulate the mutations, and that is why we have yearly flu shots, not a single vaccine. Every year we continue to monitor which type of these flu viruses are circulating in our surrounding, and then on the basis of the most commonly circulating viruses, we design the vaccine for that particular year. Type A viruses are classified into 18 distinct H types and 11 distinct N subtypes. Flu viruses can also undergo genetic recombination, so it means that when two subtypes infect the same cell, so within the same cell environment, the mutation chances are more. So that's why they create novel combinations of spikes that are unrecognizable by the human antibodies or the vaccine that has been already entered the human body. So in that case, they might cause the severe infection. So if you see here the one of the previous pandemic, what happens in 1918, it is famous as Spanish flu. It killed about like 50 to 100 million individuals worldwide. It is it is said that it was the type A influenza virus that has H1 and N1 spikes. So the type A influenza virus that has the subtype H1 and N1. Then the second one that was another very common or very famous one is that happened in the 1957 Asian flu. It has a same subtype A, but sorry, the serotype A influenza virus, and the subtypes were H2 and N2, and it killed about 100,000 Americans. After that, in 1968, we have Hong Kong flu. It infected about 50 million in US, and it killed about 70,000 individuals in US. The type was again influenza type A, while the while the subtypes were H3, that is hemoglobin agglutinin type 3, and the neuraminidase type 2. So all these, how these are happening is that you can see here that we have these H antigens and the N antigens. One type that enters in one human, and at the same time there is another type enters in the same human, and they find a same area where they are transcribing, where they are multiplying, where they are making their proteins. At that time, what happens? The mixing of the genes and the genomes take place, and the maximum chances of mutation that results in the development of new strain that have more severe type of infection, because we do not have any previous antibodies available for that particular strain that has been developed, and that is why these new strains becomes more difficult to treat. So they mostly originate in the Far East. Viruses, the common hosts for these viruses are ducks, chickens, and pigs. In Asia, often they live in close proximity to each other and humans, and that is why the simultaneous infection with different strains favors the genetic recombination. So the new mutations occurs and new strains might develop. In 2009, there was another pandemic, that is H1N1. It was because of the viral reassortment of avian, human, and swine viruses. So, like somewhere, all three viruses came together in same host. As a result, a new species was developed that carries the genetic characters from the birds, from the humans, and from swine. And as a result, the death toll was similar to seasonal influenza, but more severe, or the mostly the younger people were more affected by that. Now we have another term, that is emerging viruses. So the viruses that originate in one organism then pass to another and causes the disease. So this current coronavirus outbreak is an example of the emerging virus, because it is said that this coronavirus basically was found in the bats, and then might be it jumped into some intermediate host, and from that intermediate host, that is most likely the people or the papers are coming that it could be a pangolin, that is a ant-eater, scary ant-eater, and from that it jumped into the humans. So these emerging viruses are often deadly to new hosts and considerable threat in the aviation age, because viruses do not have any boundary. They can stuck to different surfaces and then move with the humans from one place to other place, and that's how they can spread so quickly, and this is what has happened in the case of coronavirus infection. Another example is, for example, hantavirus. It causes deadly pneumonia. Natural host is deer mice, and simply controlling the deer mice has limited the disease. One more example of these viruses is Ebola virus. It causes severe hemorrhagic fever. Among most lethal infectious diseases, lethality rate is reported more than 90% in outbreaks in Africa. Latest outbreak was in 2014, end of the 2014, and still we are having some cases positive from that particular region. The natural host is still unknown. In 2003, we had SARS coronavirus, that is severe acute respiratory syndrome. It was also pneumonia-like symptoms, and it was fatal in 8% of the cases, caused by a member of a coronavirus family. Again, mutation is one of the important cause in those viruses, although it was lesser than the HIV's or the HIV virus rate of mutation, but still because of this mutation we were not able to catch that virus and control that so quickly. Its vaccine has been currently uh, is being developed. Then if you look for the coronavirus infection, so what you can see is that how does your respiratory system become a target. So when the coronavirus is introduced into the body, it enters the respiratory system, causing inflammation of the lungs, followed by inflammatory response syndrome, and in severe cases, organ failure. Mostly the cardiovascular system, immune system, and respiratory system are the ones that are affected. After being exposed to the COVID-19, and the incubation period is between 4 to 14 days, the virus travels to the lungs and causes damage to the alveoli, air sacs especially, that allows the gaseous exchange. That's why it causes difficulty in the breathing. That leads to the hypoxia, that is insufficient amount of the oxygen, making it hard to breathe, and severe coughing. Next, the infection can enter the bloodstream and spread throughout the body, causing your body to have a high fever. And that is why some of the people who are having problem with this, they are having multiple organ failure also. There are a variety of viruses that can cause the cancers. They may contribute to about 15% of all human cancers. Viruses can cause cancer by altering the growth properties of the human cells, for example, by triggering the expression of the oncogenes, or disturb the cell cycle or the growth cycle, controlling genes. So in 2006, FDA approved use of a HPV vaccine to prevent cervical cancer, and it is recommended for both male and female 11 to 12 years old individuals. So it is just an effort to control the cancers that are related to the human papilloma virus. Beside viruses, there is another term that we are hearing now is prions. So what these prions are, these are basically abnormal proteins, or in particular proteinaceous infectious particles. What they do, they cause transmissible spongiform encephalopathies. And both humans and animals can experience the disease. With reference to the animals, very famous one is the mad cow disease or bovine spongiformis encephalopathy. In sheeps, you can see scrapie, and in humans, the most famous one is the Creutzfeldt-Jakob disease or CJD. Uh, Normally, the host has normal prion proteins, and that is PRPC, and um, these abnormal proteins that are PRPSC, they causes the normal PRP to get misfold, and then it can cause disease. Normal PRP protein is necessary for the productive infection by scrapie infectious particles. So here you can see this is a scrapie infected mouse that has these defected proteins. Its brain be homogeneous, and it was transferred into the PRP deletion mouse. The mice live normal lifespan and brain appears normal. But on the other hand, the control mice that does not have the deletion of the PRP, when it was injected with this infected mice proteins, the mice die of neurological disorders because the PRP converted into PRPSC, and the brain section shows the characteristic damages. So what happens in case of the mad cow disease, for example, that all those individuals or the people who consume the brain and the meat of those infected bovines or those cattle or those cows, they got infected and they showed the symptoms of this particular disease. Now, when we have viroids, so what viroids are? These are the tiny naked molecules of circular RNA. So no capsule, no capsid, no envelope, only the circular RNA, and it can cause disease in plants, or you can say it is more known that with reference to the viroids that they infect the plants. So for example, recent outbreak killed over 10 million coconut palms in the Philippines. It autonomously replicate and info appears to be in 3D structures and not the RNA. So we might use the plant um, siRNA machinery to affect the gene expression. Here you can see there are some of the common viral infection. For example, chickenpox. It is caused by varicella zoster virus. It has a double-stranded DNA. It spreads through contact with infected individuals. No cure is available. It is rarely fatal, and vaccine is available. On the other hand, hepatitis B, another viral infection caused by papillomaviruses, double-stranded DNA viruses. Again, vaccine is available. Herpes is caused by herpes simplex virus, double-stranded DNA, and blister spread primarily through skin-to-skin contact with cold sores and blisters, very prevalent worldwide, and again the vaccine is available. Mononucleosis, or in layman word, just the mono, caused or infectious mononucleosis caused by another virus, Epstein-Barr virus, double-stranded DNA. Again, we have is very common, no cure is available, and infection may last for several weeks. Smallpox, another double-stranded DNA virus, variola virus causes this infection. AIDS virus is a caused by HIV virus, positive single-stranded RNA, and then the polio enterovirus that is caused by RNA virus. West Nile virus is caused by flavivirus. Ebola caused by again viruses, negative single-stranded virus. No vaccine available. Influenza, negative single-stranded RNA virus. Influenza viruses, vaccines available, and they changes every year due to the depending upon the whatever the yearly circulating viruses are. Measles, caused by paramyxoviruses, group of paramyxoviruses, and the virus has negative single-stranded RNA. SARS, negative single-stranded RNA, and rabies caused by retrovirus, single-stranded RNA. So here we are closing this part 2, and after that I will be continue with the part 3 of this lecture series. Thank you very much.